So the title of Dr. Hagstrom's talk is Tulp1 Missense Mutations Cause Variable Retinal Phenotypes.
Thank you, and I apologize for the technical glitch, and thank you for this invitation and opportunity to talk about one of the research projects that are ongoing in my lab. And I cannot advance the slide, man. Okay, thank you. So my team studies the mechanisms of inherited retinal diseases, which is highlighted by progressive photoreceptor degeneration, namely the rods and the cones. And there are different forms of inherited retinal diseases, such as retinitis pigmentosa, Leber congenital amaurosis, and cone rod dystrophy, each of which can be caused by different genetic mutations, and these can initiate at different times in a patient's life and can progress at different rates in different patients. So what generally happens is that the rod photoreceptors in the periphery degenerate, and then the degeneration progresses so that they only have a central island of vision left that's shown here down on the right. And eventually this area too will degenerate and patients will lose all of their vision. Surprisingly, there are thousands of mutations in over 200 genes that can cause these different forms of retinal degeneration. And this heterogeneity is extremely challenging with regard to finding treatments and therapies. So one of the primary goals in the field is to not only identify mutations and to identify the functional consequences of these mutations, but very importantly, to identify the disease mechanisms in order to target specific pathways to generate therapies to slow or halt the vision loss. Next slide, please. My group focuses on a gene called Tulp1, which when mutated causes early onset severe forms of retinitis pigmentosa and Leber congenital amaurosis. And so I'm going to use this as an example for understanding disease mechanisms. Tulp1 is a photoreceptor specific protein that's shown here in green on the right retinal section, and it localizes to specific photoreceptor compartments, namely the inner segment, the connecting cilium, and also the synapses. To study the effects of Tulp1 mutations on the retina, we generated a knockout mouse model. So these animals lack a functioning Tulp1 gene. And when we analyzed the retinas of these mice, we were happy to see that these Tulp1 knockout mice show an early onset progressive photoreceptor degeneration, similar to what's seen in human RP patients. And this is shown in the panel on the bottom right, where by 3 weeks of age, these mice are already starting to show photoreceptor degeneration with slightly less nuclei. You can see the nuclei count is down here, and some disorganization within their outer and inner segments. And by 8 weeks of age, hardly any photoreceptors are remaining. We also noted several other phenotypic features in this model that's not shown in the slide, but is relevant later in this talk. And that includes the mislocalization of several outer segment proteins and the synaptic termini lacking their tight spatial relationships between synaptic ribbon proteins. Next slide, please. As I mentioned, what's commonly done to study gene function is to generate a knockout model in which the entire gene of interest is eliminated from the mouse genome, and then subsequently the mouse model is studied. Within the past several years or so, there's been a game-changing technology in genome editing called CRISPR-Cas9 technology, and this has really revolutionized biomedical research. CRISPR-Cas9 is a specific, efficient, and versatile gene editing technology that scientists can use to modify, delete, or correct precise regions of DNA. And in the study I'm going to highlight today, we use CRISPR-Cas9 technology to better recapitulate human RP patients' gene dosage and spatial temporal degeneration. And we generated 2 what we call knock-in homozygous mutant mouse models, each that express a human RP causing Tulp1 missense mutation. So these mice express a mutant Tulp1 gene instead of completely lacking the Tulp1 gene. And then we went on to ask the questions: how do these new models compare to a knockout mouse? And how do they compare to the wild type retina with regard to the development of photoreceptor degeneration? And do the photoreceptors die via the same disease mechanism? Throughout the rest of the talk, I'm going to refer to these new knock-in models as D94Y and F491L, and that indicates the location and the amino acid change in the gene. Next slide, please. The first experiment we did was to determine the distribution of Tulp1 in these transgenic mice. And this is immunohistochemistry immunohistochemistry showing the retinas of these mutant mice, and in green is the expression of the Tulp1 protein, and we can see that in the wild type mouse, it is expressed in the inner segments, the connecting cilium, and the synapse. And it's also shown in the exact same compartments in our mutant retinas. And it is not expressed in the knockout model, which is what we expected. And these results indicate that the retinal distribution of the Tulp1 protein is not grossly affected in these mutant retinas. Next slide. Next, we studied the retinal morphology of these mutant mice using light microscopy at multiple time points. For simplicity, I'm only showing 2 time points here to illustrate our results. At postnatal day 17, all 3 mutant retinas show relatively normal photoreceptor or retinal morphology, where they have a full complement of photoreceptor nuclei and organized outer and inner segments. But by 2 months of age, the F491L mutant and the Tulp1 knockout have a degenerating retina where they only have 1 or 2 rows of nuclei remaining, and their inner and outer segments are completely gone. Whereas in the D94Y retina, it is very similar to wild type. So these initial results suggest to us that the disease severity varies with genotype in these different Tulp1 models, something we would not have known without generating these knock-in models. Next slide. To test the retinal function, we performed electroretinography. And again, I'm only showing data from 2 time points. So this slide shows a series of dark adapted ERGs. At P17, there's an overall reduction in the Tulp1 knockout response that's shown in red. And at the same time point, the ERG was also reduced in amplitude in our F491L mutant that's shown in blue, as compared to the wild type, which is shown in black. Both the A and the B wave reduction progressed with increasing age, that's shown in the P60 time point. And this was consistent with what we saw in the anatomical features shown by histology in the previous slide. So again, it shows that our F491L mutant is degenerating similar to the knockout, and the D94Y is not. Next slide. Next, we analyze the localization of several outer segment specific proteins. Previous studies show that outer segment proteins are mistrafficked in the Tulp1 knockout retina. And so panel A here shows the expression of rhodopsin, the molecule that grabs the photons of light. And it is correctly trafficked in the wild type and the D94 mutant. It is supposed to be expressed specifically in the outer segments, as it is here. But in the F491L mutant and the knockout retina, it is expressed throughout all the different compartments of the photoreceptors. To examine the cones, we immunostain the retinal sections with antibodies against the cone opsins, the S opsin and the M opsin. And these panels show that these opsins are also correctly trafficked and retained in the outer segments of the D94Y and wild type, but they're mislocalized throughout all of the photoreceptor compartments in the F491L mutant, similar to the wild type. Next slide. We next focus on whether or not these Tulp1 mutant retinas revealed photoreceptor synaptic defects, which is something we saw in the knockout retina. And we examined these synapses, their architecture, using antibodies against ribeye, which is a protein that is a central scaffolding protein of the synaptic termini. So ribeye normally localizes at the presynaptic membrane in a horseshoe shaped, and you can see this with the white arrows in the wild type retinas. And this is similarly seen, these horseshoe shapes are similarly seen in the D94Y retina, but in contrast, the 491L retina and the knockout show a very thin and punctate staining, and normal shaped ribbons were rarely seen. Next slide. So to investigate whether or not these D94Y mice actually go on to develop photoreceptor degeneration, we evaluated these mutant retinas at a very late time point at 12 months of age and 18 months. And right here, I'm showing you the data from 1 year. Histological analysis shown at the top clearly shows that there's minimal changes between the D94Y retina compared to wild type. There appears to be some slight less pigmentation in the RPE cells and the presence of a few dilated structures in the synapses that is shown with the black arrowheads. These dilated structures in the synaptic layer led us to evaluate these mice via ERG. And the ERG reveals, shown on the bottom here, that the A wave is comparable to the wild type in the D94Y mice. but the B wave is slightly reduced under both the dark adapted and light adapted conditions, indicating a defect in the photoreceptor to bipolar cell neurotransmission. Next slide. So now, having identified that there are these differences across our Tulp1 mutant models, our goal was to identify the mechanism of disease, which may be different in these models because they actually have different phenotypes, something we didn't expect. Recently, many studies have examined the involvement of the endoplasmic reticulum unfolded protein response pathways, otherwise known as ER-UPR, in neurodegenerative diseases, including Alzheimer's, Parkinson's, Huntington's, all of which have these heritable mutations that cause accumulation of misfolded and mistrafficked proteins, similar to what we see occurring in the photoreceptors of our Tulp1 knockout and F491L mutant mice, with the rhodopsin and the conopsins being mislocalized and accumulating throughout all the photoreceptor cell compartments. So evidence indicates that these misfolded and mistrafficked proteins can initiate a pathological process that disrupts the ER quality control pathways, leading to ER stress and eventual neuronal cell death. And these UPR pathways are what's shown here on the left. There's many different pathways, and the details are not what's important.
Stephanie, can you wrap it up, because you've passed your time?
So we went on to look at some of these markers, and we can see that these markers are much more highly expressed in the F491 mutants than they are in the D94Y mutants, which is what we were expected to see. Next slide. So the conclusion from our study was that we wanted to evaluate the pathogenicity of the different Tulp1 mutations, and we generated these two novel mouse models, each expressing a different RP causing mutation. And these phenotyping experiments indicate that the two mutations cause variable retinal phenotypes. And that when probing for a specific mechanism, we saw that the ER-UPR pathway is activated in the F491L model, but not the D94Y, highlighting the fact that different mutations in the same gene may cause cell death due to different disease mechanisms and may then require unique therapies. Next slide. So these studies form the basis for future directions where we want to probe for additional evidence of ER-UPR activation. And then most importantly, we want to analyze the effect of known ER-UPR modulators as pharmacological therapy to then delay or slow down the development of photoreceptor degeneration. So if we can highlight some of these upstream pathways, we're hoping to, with pharmacological therapy, hit these pathways at different levels and try to delay or slow down photoreceptor degeneration. Next slide. And finally, I'd like to acknowledge the people of my lab, past and present, who have been part of these projects, our Coli Institute collaborators that helped with scientific input and technical expertise, and of course, the funding resources from the NIH, the Research to Prevent Blindness, and Cleveland Eye Bank Foundations for supporting my work. Thank you very much.
Thank you, Dr. Hagstrom.